To start with, blood clots are when blood changes from a liquid to a solid. So first type of blood clot is thrombosis, which it's stationary, doesn't move, can block blood flow. A series type of thrombosis is DVT, when, your deep veins clot in your body, which deep veins carry a majority of your blood, and, obstruction of those can be life-threatening. Then you have superficial venous thrombosis, which is the inflammation and clotting in a superficial vein, which a superficial vein is a vein that is closest to the surface of your body, which are usually veins that you're able to see like while you're lifting something heavy. Then we get to pulmonary embolisms, which like a DVT could turn into an embolism. What an embolism is it's a blood clot that breaks loose from a blood vessel and can travel around the body. So it all starts with a cut on the skin or an internal injury, which causes a tear to a blood vessel wall, causing blood flow. Then we get to vessel constriction, which is when blood vessels narrow to control blood loss or limit blood flow. Next, you get this thing called a platelet plug. This is when platelets are activated in response to injury. The platelets stick together in the wound site to form a plug. And finally, we get to a fibrin clot. This is when blood clotting factors trigger the making of fibrin. The fibrin is a strand-like substance that will surround the platelet plug and keep the plug nice and stable. This is what's called a fibrin clot. The clot then gets stronger and then will finally dissolve as the blood vessel heals. The proteins that help this process happen are called the blood clotting factors. Now, the blood clotting factors are the 13 proteins that help form a blood clot. The first blood clotting factor is fibrinogen. Second is prothrombin. The third is tissue thromboplastin. Fourth is calcium. Fifth is the labile factor, but it can be canceled out by another factor. The sixth is unknown. Seventh is the stable factor. The eighth is the anti-hemophilic factor A. The ninth is the Christmas factor. The 10th is the Stewart power factor. The 11th is the plasma thromboplastin factor. The 12th is Hagman factor. And the 13th is the fibrin stabilizing factor. I found a mnemonic that can help you remember all of these factors, which goes, first person told cancer leads sickness. Another chap said protein high fat. Now, after all of that, let's get on to the lab itself. I'm going to put on some gloves because this lab has the potential of being messy. And these are just some regular vinyl gloves. They're not latex because I couldn't find any. Next, I'm going to get some distilled water in a measuring cup. I am then going to fill that measuring cup to about a quart. While this fills up, I'm going to give you a random fun fact. Did you know if you lined up every blood vessel in a toddler's body, that it would be up to 60,000 miles, and an adult 100,000? Now we're ready for our blood mix, which is a weird powder. So I'm going to use this knife to cut this plastic open because it was really hard to cut open with my fingers. So I decided to use a knife. I just used it for slicing the cap, and then I opened it with my hands. Then I poured the water inside of the artificial blood. It took a little bit because I was kind of worried that the blood was going to get everywhere and my carpet was going to be stained. That would have been bad, but fortunately I managed to do it. So now I'm going to put the cap on the bottle, make it nice and tight, and then shake. Shake it for a pretty good bit, cut it because it was taking too long. Now I take my gelatin strips and some scissors, and I cut the gelatin strips into little strips. So I do this, I just cut these little half strips, and then I cut sideways, so they just all come off. They're all gonna fall on this piece of paper towel. Then I will get my kidney dish, my first kidney dish, and then put all the gelatin strips inside of this kidney dish. Then I will get my second kidney dish, forceps, the fake blood, and a plastic spoon to mix. Now I'm going to pour my fake blood into the second kidney dish. Fun fact number 2, blood in the veins or deoxygenated blood is not blue. It is actually a darker tinge of red, like maroon. It amazes me that there's still people in my grade that still think blood is blue. It's just funny to me. Now we get on to the whole reason why we're here, the ending result of this lab. So I'm going to get my gelatin strips. After I get these forceps and a plastic spoon out, I'm gonna get all my gelatin strips. I'm gonna pour them into the fake blood. Make sure I get all of them in there. All right, now I'm going to mix with my plastic spoon. I'm just gonna mix them around. They eventually disappear, or look like they disappear. Now, what they create is a blood clot type structure. Now you see the activated gelatin strips. Their texture changed a little bit. Again, the color of the blood, feeling changed.
I got one.
You see me get this pretty good one. I want to show this one that I found to the camera because it was a pretty decent one. So I'm going to start scooping out the blood clots or the gelatin strips. I'm going to get them on this kidney dish so I can show them. You can see how good that blood looks. I would say the things that I learned while making this project made this really fun, and I would love to do something like this again.